Hello and welcome to my channel Lab Easy. So in my previous video, I had discussed about the procedure, the step-by-step procedure of performing a Western blot. And today, I will be discussing what went wrong in your Western blot. So in this video, we will discuss the problems that may arise in your Western blot and which are usually very common. So getting a perfect plot in the first time can be very tricky and there might arise several problems. So we will discuss about them and we will discuss the probable solutions by which you can improve your plot. So let's begin. So I will go step by step. The first step is separation. As I had discussed in my previous video also, please check out the link below and see my previous video here. So in the step of separation, that is, our protein samples are being separated according to their molecular weight. So what may happen is that if your protein is of very high molecular weight, it may not be very well resolved in your gel. So what may happen is that according to your gel percentage, your proteins may not be well resolved. So a probable solution of it is that you may change the percentage of gel. And gel percentage, that means here I'm talking about SDS page. So in this case, we treat the proteins with SDS as I had discussed in my previous video also. And the proteins are denatured and they are imparted with uniform negative charge. So the proteins migrate according to their molecular weight. Now, a gel percentage is also very important in this case. And what is the case here? That if the gel is of higher percentage, so usually we use a 12% SDS page. And in that gel, the pore size is defined, the pore size between the polyacrylamide. But if you reduce the gel percentage, so if we make it 10% gel, then the pore size is increased. So higher gel percentage usually means a lesser pore size. And we can also reduce the gel percentage to 8%, which will again increase the pore size of the gel. So if your protein is a very high molecular weight, for example, 200 kilodalton, 250 kilodalton or so, then you might have to increase your gel percentage, reduce your gel percentage. That is, you can use 8% gels so that your pore size is higher and the proteins can be well resolved. So this is very important because if your proteins are not well resolved, they will obviously not give proper bands on the blot as well. And if your protein has migrated very less in the gel, for example, like this protein, if it is very close to the well, if it remains very close to the well, then what happens in the blot, it is not transferred well, and so it might not be detected in your blot. So that's the first step. Next step, what may occur, what may go wrong in the next step, that is transfer. Now we will discuss. So in case of transfer, the very first thing that we have to keep in mind is we have to place the gel and the membrane so that there is no bubble inside the gel and the membrane. Why is it so important? Because if there is a bubble, the electric current will not pass through the bubble, and which will cause an improper transfer. Because if there's a bubble on your protein band between the gel and the membrane, your protein will not be transferred properly on the blot, and which will, in the blot, it might come like this. Like there will be a bubble which you may observe on the blot as well. So please roll over the membrane. So we can use a roller, or we can also use a tube for rolling over the membrane so that we can remove the bubbles after placing the gel. Next is, suppose your protein ladder is not visible after transfer. So in case of Western blot, the most commonly used protein ladder is a pre-stained protein ladder. So in this pre-stained protein ladder, there are many proteins which have particular stains. So they can be observed on your gel, as well as when this ladder is transferred onto the blot, they can be also observed on the blot. So according to the uh, presence of this protein ladder, the pre-stained protein ladder on the blot, you might able to be confirm that the transfer was complete. And if this protein ladder is not visible on your blot properly, what might happen? One thing is that the transfer did not happen at all. So how that probability might arise is that while doing a transfer, we have to place the membrane towards the positive side of the electrode. Why? Because our proteins are negatively charged and they travel towards the positive electrode, which is the anode, right? And this is very important for transfer of the proteins, right? And sometimes we all have faced a problem like this, that we have placed the membrane on the wrong side of the electrode. That is, so what is the usual case? What is the right case here? That is, the membrane is placed towards the positive side and the gel towards the cathode side. So while we are placing this transfer cassette inside the transfer tank, we should keep this in mind. And if the case happens like this, that the gel is placed towards the positive side, then the membrane, then the protein which is negatively charged will not be able to go towards the positive side because they are on the positive side itself, so the transfer will not occur. So please ensure that the membrane is placed towards the positive side of the electrode. And one hack that you might try is Ponzo stain. So you might also use this Ponzo stain, which is water soluble stain. So this is used to stain the blot after transfer. So if your blot has all the proteins after the transfer or not, you can check it by a Ponzo stain. So it's a very easy method. You dip the blot in a Ponzo stain, and after some time you will be able to see whether your proteins, you will not be able to confirm whether your protein is transferred or not, but you will be able to see some proteins on the blot, and by that way you will be able to confirm that yes, the transfer has at least occurred correctly. And after observing these proteins on the blot, what you do is you place this membrane in water so that the Ponzo stain is washed away. So that's a very easy method. Okay, coming to the other problems that might occur during transfer, the one that occurs very frequently is that if you're transferring for a long time, so usually transfer takes 30 to 60 minutes. But if your protein is, suppose, of high molecular weight, you might want to do transfer for some longer time. And one problem that may arise here is that during the transfer, a high electric voltage is being applied, and in that process, the gel may overheat and it may melt. So if your gel is molten, obviously your proteins, like the transfer of the proteins, will be hampered. So that is why you should always carry out the process of transfer in lower temperatures. So we usually carry out transfer, for example, in 4 degrees centigrade, so inside cold room or in a lower temperature. That is one thing. Another is that like if your transfer is not occurring correctly, there are many things that you can check. One is your transfer buffer components. So I had discussed in my previous video as well. Transfer buffer contains stress, glycine, and methanol. So check your components properly, whether they are correct or not, and then prepare the transfer buffer freshly so that your transfer can be carried out easily and smoothly. And another thing that you have to standardize in this whole process. So actually, Western blot requires many standardizations. We will see in our next detection step as well. And here in transfer step also, you need to standardize the transfer time. That is, for how long will you allow this transfer to occur so that your protein is transferred properly and it will be visible on your blot. So that's for the transfer. Next, we will move on to the detection step. So in case of this detection step, there are many things that might go wrong, but don't worry, there are solutions. So first thing is, suppose your band is not visible on the blot. What may go wrong is that your protein amount might be very low. So if your protein amount is low, it will not be easily detected by the antibody. So if the antigen is low, then this antigen antibody reaction will not be properly occurring. So of course, because of that, you will not be able to see your band. So to rectify this, you have to measure your protein amount and you have to standardize how much protein you will load. And for protein amount measuring, we usually take help of Bradford assay. So by Bradford reagent, which uses colorimetric detection, colorimetric measurement, we can measure the protein amount that we are loading into the gel so that we can ensure that a proper amount of protein is loaded and it can be detected by our antibodies. One very, very common problem that occurs in blots is that the background is very much like you can't see anything on the blot, everything is black. These things may happen. So there are several possibilities which might occur here, which is, so before you detect, so there is one step which is blocking, right? And if the blocking is not done correctly, what do I mean by that? That means, suppose it was done for a very short time. You need to increase the time of blocking. You need to check which blocking reagents you are using. If you are using skimmed milk or if you are using BSA. And if you are using BSA, you should always take a fresh aliquot of BSA so that blocking can be done properly. Next is wash. Wash is very, very important in case of Western blot, and wash should be done after blocking as well as after antibody incubation. So if you're done, if you're doing less, like if you're doing wash, so if you're washing the blot for less time after blocking, then what may happen is that your blot might still contain excessive amounts of the blocking reagent, which may hamper the interaction of your antigen and antibody. So that is why the wash should be done properly. And also after you incubate with primary and secondary antibodies, such that there is no excess amount of antibodies which are remnant on the blot. Next thing, which is also essential in this to reduce background, that is incubation time with antibody. Sometimes we have seen that if we are incubating with the antibody for a very long time, suppose if you're incubating with the antibody overnight, there may arise background. So to prevent that, we have to standardize the incubation time of the antibody. And this also depends on the types of antibodies, which I'm going to discuss later. Okay, now another thing might happen is that non-specific bands may also arise. So non-specific bands means bands of proteins which are not your desired protein, but some other proteins. So this requires a lot of standardization to reduce your non-specific bands. The first thing is dilution of antibody. So what I mean by that is that from the stock, we usually make a dilution. So suppose we can dilute the antibody in this way, like 1:500 dilution, 1:1000, 1:5000, and so on. So along with your antibody, there will come a sheet, a technical sheet, where it will be mentioned how much antibody, how much dilution is usually preferred for blots if you're using whole cell lysates. And that also you might have to standardize a little bit. So usually for primary antibodies, lower dilutions are used. 1:100, 1:500. These are usually used for primary antibodies. So primary antibodies are used in lesser dilution, but secondary antibodies might be used in like higher dilutions, like 1:1000, 1:5000. So more dilute secondary antibody aliquots can be used. So this antibody dilution you have to take care of. Next thing is incubation time. So as I've already mentioned in the previous point as well, um, you have to standardize how much time you're incubating with primary or secondary antibody. Next is, it also depends on the type of antibody that you are using. It may be monoclonal or it may also be polyclonal. So uh, these things you have to play around with. If your antibody is monoclonal, it might require lesser time, lesser incubation time. So this has to be considered. Next thing is antibody specificity. Are you using the correct antibody at all? So here, if like we use two antibodies, right? The primary antibody and the secondary antibody. So the primary antibody is specific to your antigen, but it is raised in a particular organism. Suppose your primary antibody is raised in rabbit. So if it is raised in rabbit, your secondary antibody has to be an anti-rabbit antibody. That means your secondary antibody has to recognize the constant region, that is the FC region of rabbit antibodies, rabbit IgG, for example. So your secondary antibody has to be specific for your primary antibody. So if you're using antibodies which both are raised in same organism, like both are suppose raised in rabbit, then of course your secondary antibody is not going to work. So your antibodies, the primary antibody and the secondary antibody, has to be raised in different organisms. All right, and so that's why you have to check whether you are using proper antibodies or not. Next thing that might happen, that you do not have any background, you do not have any other problem, your transfer is also proper, but you still do not see your desired protein band. So this is the process of detection shown here. And you can see here that this is the antigen, the protein, the target protein. This is primary antibody here in green. And this one is secondary antibody, which is conjugated with an enzyme. Right? So if your desired protein band is not observed, you have to check whether you are using the proper secondary antibody or not. Whether this enzyme, which is conjugated with this antibody, is this enzyme working properly? That also you have to check. So here, in case of Western blot, the mostly used detection technique is chemiluminescence. I had discussed this in my previous video as well. And in case of chemiluminescence, the mostly used enzyme is HRP, horse radish peroxidase. So there are substrates which are luminol and H2O2, which are used by this HRP to give chemiluminescent signal. So you have to check whether this enzyme is working properly or not. So you have to, in case in any way your protein band is not visible, then you have to check with different alicot of secondary antibody to check whether whether this enzyme is working or not. And the substrates which you will be using, like luminol and peroxide, whether they are degraded or not. That also you have to consider, because H2O2 degrades if you keep it in light. In light condition, if you keep it, it degrades. So whether your H2O2 is working properly or not, that also you have to consider. One other problem which may arise is the blotch. Like your blot may you may observe a blot like this. And I know this is very disheartening, but you have to standardize a lot of things here. That is your protein loading amount, whether you're loading right amount of protein or not. If you're loading too much of protein, then these kind of blotches may appear, where like too much of your protein is present and your antibody is giving too much of signal. So you can avoid that by reducing the protein loading amount. And you can standardize the washing time and the incubation time with antibody as well to reduce the occurrence of these blotches. So to summarize this thing up, check whether your protein is well resolved in gel, whether your transfer is complete or not. Please avoid any bubbles. Roll over the membrane so that the bubbles can be avoided. Place the cassette in right orientation, that is the membrane towards the positive side of the electrode. Block the blot properly, and so that your non-specific blotches or anything non-specific can be avoided. Wash the blot properly and check your antibodies. So that's all for this video. I hope your blotting problems can be solved. And if you like my video, please like, share, and subscribe. Thank you.